In part 3 of our series on adaptive immunity, we're going to discuss T cell activation. Our lymphatic system is what drains tissues and brings antigens into secondary lymphoid organs such as lymph nodes. These lymph nodes are connected by lymphatic vesicles. They drain interstitial fluid into lymph nodes. Once the interstitial fluid enters into the lymphatic vesicle, we call it lymph. T cells and B cells are activated as foreign antigens are brought by lymph into the lymph node, and then the lymph re-enters circulation via the thoracic duct or right lymphatic duct. And thus, this is a one-way movement. Plasma goes to interstitial fluid, which goes to lymph, which goes back to plasma. There are a number of important structures within the lymph node. This is the site that drains tissue antigens, and lymph arrives into the lymph node via the afferent lymphatics and leaves via the efferent lymphatics. But of all these structures, I really only want you to focus on the B cell zones and the T cell zones. The B cell zones are composed of primary B cell follicles. These are where naive B cells reside, as well as germinal centers, which is where the activated B cells reside. The T cell zone surrounds these B cell zones and is called the paracortex. This is a picture of the B cell and T cell zones within the lymph node. Here's the primary follicle, which is naive, non-activated B cells, and here's a germinal center, which is the site of B cell activation. These are surrounded by the paracortex, which is rich in T cells. The lymphocyte structures in the spleen are very analogous. The spleen drains blood antigens and is composed of a red pulp and a white pulp. I'm not going to get into the specifics of spleen anatomy other than to say that the white pulp is what contains the B cell zones and the T cell zones. And again, the B cell zones are composed of primary B cell follicles and germinal centers. But in this case, the T cell zone is called the periarterial lymphoid sheath, or PALS. And this is a sheath of cells that surrounds arterioles and is composed primarily of T cells. Now, we focused a lot on the inflammatory response in our innate immunity section, but now I want to turn our attention to another cell type that's present in inflamed tissues called the dendritic cell. A dendritic cell is a phagocyte, and a phagocyte can take up any pathogens that are present in an infected tissue. Dendritic cells may also be infected directly by things like viruses. But when a dendritic cell is exposed to these pathogens, it's also exposed to the inflammatory environment, either via its own pattern recognition receptors or via inflammatory cytokines that are in that environment. And when that happens, the dendritic cell becomes activated. It increases its ability to present antigens, and it upregulates chemokine receptors that allow for its trafficking to the draining lymph node. Once it gets into the lymph, it will carry those antigens that it has taken up to the draining lymph node and present them to T cells. And bear in mind, the draining lymph node the lymph can also drain all sorts of other things, such as pathogens themselves or pathogen products, and carry them to the draining lymph node. Now, once within the draining lymph node, dendritic cells can activate T cells and mediate their activation and initiate the ability of those T cells to undergo a process called clonal expansion. So what is clonal expansion? Well, the frequency of naive lymphocytes that might be specific for any given antigen is actually quite low. Remember, our antigen receptor repertoire is extremely diverse. In practical terms, it's probably about 1 in a million lymphocytes that might be able to recognize any specific antigen. But when those lymphocytes recognize that antigen, they undergo a process of cellular division, which we call clonal expansion. This increases the frequency of T cells specific or B cells specific for that given antigen from 1 in a million up to 1 in 50 or 1 in 20 or even about 1 in 10. This means that lymphocytes in the course of about a week can undergo about 20 cell divisions, which is about as fast as mammalian cells can divide. So what are the signals that induce this cell division? Well, first, I want to talk about signal 1. Signal 1 occurs when a T cell receptor binds to an MHC presenting a peptide. But there's 2 other components of that signal that are important. One is the co-receptor, either CD4 or CD8 is also part of this complex that binds into the MHC. And the other key component are all of these subunits that collectively we refer to as CD3. These are the subunits that actually deliver an intracellular activation signal to the T cell when the T cell receptor binds to an MHC peptide complex. CD3 is important to know because it is the complex of molecules that's used as a marker for T cells. So a cell that is expressing CD3 is by definition a T cell. The second signal that T cells have to get activated is referred to as co-stimulation. And this signal occurs when APCs express co-stimulatory molecules when they have become activated. And the two most well-known co-stimulatory molecules are B7-1 and B7-2, which are also known as CD80 and CD86. When these bind to CD28 on the surface of T cells, these induce a co-stimulatory signal into that T cell, which enhances its activation and survival. Now, some co-stimulatory signals actually deliver a negative or regulatory signal, and this includes two important molecules, CTLA4 and PD-1. These are negative co-stimulatory receptors that are expressed after T cell activation. CTLA4 actually binds to B7-1 and B7-2, but with higher affinity than CD28, and so it competitively inhibits the ability of CD28 to bind these molecules. Both CTLA4 and PD-1 deliver suppressive signals to the T cell that downregulate their activation. You can think of it as CD28 being the accelerator on T cell activation, whereas CTLA4 and PD-1 are the brakes. And it's important to know about PD-1 and CTLA4 because these have become important targets for cancer immunotherapy. Blocking these molecules can enhance the T cell response to tumor antigens. Okay, so to summarize, T cells are activated within the T cell zone of the lymph node, where many of those T cells can differentiate into activated or effector T cells that can then migrate to inflamed tissues. In later video lectures, we will also discuss that some helper T cells stay in the lymph node and migrate to B cell zones and enhance B cell activation.